I hope I'm audible.
Yes,
So, good evening, everyone. I'm Khushbu, and I welcome all of you on behalf of Strand Life Sciences to a wonderful webinar on unlocking CAR T cell innovation, how CITE-seq is revolutionizing immunotherapy research. We have two speakers for this webinar. Our first speaker would be Raj Jha. He's the Associate Bioinformatics Engineer at Strand Life Sciences. His experience includes an internal R&D initiative at Strand. He has been actively involved in an external client project in collaboration with LV Prasad Eye Institute. He has executed numerous custom analysis leveraging the Surat package in the R programming language and has collaborated with the School of Medicine at Mount Sinai. His educational background includes his master's at IBAB at Bangalore and uh, honors in biotechnology from the Cotton University. Our next speaker would be Nihar Lakhe. He is also an associate bioinformatics engineer at Strand Life Sciences. He has worked on trio-analysis using multi-omics approach to investigate occurrence of CHD infants. He's also worked on developing and testing scRNA-seq and CITE-seq pipeline using Nextflow and NFTest, worked on manual testing and automation testing using WebDriver IO with JavaScript. His educational background includes his master's from IBAB and he has done his bachelor's from Dayananda Sagar University. I welcome both of you today and I would request you to please take the session forward.
thank you, Kintu. I'll share my screen. Am I slides visible?
Yes, it's visible.
Yes. Hi everyone. As Khushbu mentioned, today I and Raj will be presenting on the topic unlocking CAR T-cell innovation, focusing on how CITE-seq is revolutionizing immunotherapy research. Before we start our presentation, I'd also like to briefly introduce Strand. Strand is a 23-year-old omics CRO company which has 15 years of experience in curating variants, genes, pathways, and diseases for clinical reporting and pharma biotech custom solutions. It has over 22 years of experience in providing bioinformatics and software solutions. Along with that, it also has 11 years of experience in various omics assays. In today's webinar, we'll be focusing on various topics. Firstly, on CAR T therapy, we'll be presenting a brief overview of CAR T therapy with its procedure and clinical applications. We'll be focusing on CITE-seq technology and its analysis and how that would help in CAR T therapy. We'll also discuss about the design of the pipeline and its overview, how we can scale the pipeline for using CITE-seq to better provide the CAR T therapy, and other key features that can be included in this pipeline. Coming to the CAR T cell therapy, CAR T cell therapy, also known as chimeric antigen receptor T cell therapy, is a type of personalized immunotherapy where a patient's own T cells, a type of immune cells, are genetically modified to express special receptors called CARs on their surface. These engineered receptors help the T cells recognize and attack cancer cells more effectively. In order to understand the effect of CAR T cell therapy, we'll also look into the procedure of it. The procedure broadly involves collecting the patient's T cells at the treatment center, modifying and expanding them at a specialized manufacturing unit, and then infusing them back into the patient at the hospital after processing. The first step is to identify treatment for patients. CAR T cell therapy is often utilized when traditional methods like chemo and radiation fails. Leukapheresis is done to extract T cells and return other blood products. These isolated T cells are then cryopreserved and shipped to the manufacturing unit, where these isolated T cells are modified. The insertion of the CAR, which is a chimeric antigen receptor, into T cells, known as transduction, is typically done with the help of viral vectors, which forms the CAR T cells. These CAR T cells are then expanded to form many copies. These expanded CAR T cells are then cryopreserved and shipped to the treatment center. Before infusing the CAR T cells, The patient undergoes lymphodepletion. This lymphodepletion helps in making the space for manufactured CAR T cells. Finally, the CAR T cells are infused into the patient's blood. Inside the body, CAR T cells target these cancer cells. So to understand the effect of CAR T cells, we'll also need to look into the CAR protein, its structures, and what are the different generations of the CAR protein. CAR, also called as the chimeric antigen receptors, are engineered fusion proteins designed to recognize antigens on cell surfaces. As seen in figure A, we can see that the cancer cells are recognized by CAR T cells with the help of CAR proteins. CAR proteins were first developed in 1989 by Isher's group at the Weizmann Institute. CAR T cells have rapidly evolved through multiple generations, as you can see in figure B. But before we go into different generations, I'd also like to talk about the CAR protein structure. So the CAR protein consists of three main domains. First being the extracellular domain that helps in antigen recognition. Next, a transmembrane domain that helps in structural support. Lastly, an intracellular domain is an important domain that helps in T cell activation. One of the important features of CAR T cell is that it helps to kill and target the cancer cells without utilizing the HLA cells. Now we'll briefly talk about different CAR protein generations. As you can see in the figure, the green circle refers to the presence of character and red circle refers to the absence of character. As I mentioned above, that we have different generations of CAR protein 1 to 5, and all these generations have similar extracellular domain and transmembrane domain. The difference lies in the intracellular domain. However, the CD3 signaling domain is common between all the 5 generations. In first generation, we see that it only has a CD3 signaling domain. Thus, the first generation could only help in activating the T cells. In second generation, there was addition of a co-stimulatory domain. This co-stimulatory domain helps in efficiency of the T cells and their persistence. In second generation, we added proteins such as 41BB or CD28. We only added either of those to have this effect. Now, the second generation was then modified to utilize both CD22 and 4IBB that further increased its efficiency and became the third generation. Now comes the fourth generation where we saw the major change, which was due to the addition of interleukin inducer. Why was this major? Because the interleukin 2 inducer could then activate the NK cells, which are part of the innate immune system and can help in killing these cancerous cells. In the fifth cell, we see the final increase of efficiency by including two more domains, which are the interleukin 2 receptors and the binding site for transcription factor STAT3. This will allow the JAK-STAT pathway activation in the fifth generation. We'll also dwell a little into what are the clinical applications of CAR T cell therapy. So through recent studies and we have seen that CAR T cell therapy has been successful in treating ALL, BALL which is B cell acute lymphoblastic leukemia, and CLL. This CAR T cell therapy acts by targeting certain antigens such as CD19, CD20, CD22 and CD30, which are often shown by some of the leukemias. Studies have also been going on to see the effect of CAR T cell therapy on solid tumors, especially in pediatric brain tumors. Now coming to the crux of this webinar, we'll be talking about CITE-seq technology. The aim or reason behind using CITE-seq technology is to assess and validate the CAR T cell therapy efficiency. We have also mentioned below different papers and studies wherein CITE-seq technology has been utilized in CAR T cell therapy. So what is CITE-seq technology? CITE-seq, also called as cellular indexing of transcriptomes and epitopes, allows simultaneous measurement of RNA expression and surface protein levels in single cell. This addition of protein level information to single cell RNA-seq dataset is the major importance of using CITE-seq over single cell. Coming to the outputs of CITE-seq technology, so we get mRNA count of each gene for each cell, and the ADT binding count of individual surface protein of each cell. Okay, so before going into the complexities of CITE-seq technology and analysis, we'll also talk in brief about the wet lab techniques involved in this. So, firstly, what is ADT? ADT, also called as antibody-derived tag, is a combination of antibody and an oligonucleotide. The oligonucleotide consists of a PCR handle, an antibody barcode, and a poly A tail. The poly A tail present in ADT is very similar to a poly A tail which is present in your mRNA. So, once we have the ADT, the next step is to bind these cells with the desired ADT pool. In the next step, the ADT labeled cells are encapsulated within a droplet as a single cell with DNA barcoded microbits. Within a droplet, the cells are then lysed to release both ADTs as well as the mRNAs. These released mRNAs and ADTs can then hybridize to RT oligos. The RT oligos are present in the microbits. The structure of RT oligos, it consists of poly T tail, which can go and bind to the poly A tail. It consists of cell barcode, which will help in distinguishing different cells. It also consists of a UMI that helps in distinguishing PCR duplicates from real duplicates, and also a sequence adapter. Once the mRNA and ADT hybridize to the RT, they are processed for reverse transcription and we get cDNA. The next step is user specific, in which cDNA from mRNA and ADT can be separated based on size. Once the library is prepared, amplification is done independently. Amplified libraries are pooled and sequenced. So after sequencing, we'll get an RNA-seq FASTQ file and a site and a ADT FASTQ files. Further, these FASTQ files can be analyzed by us, and we can also create a pipeline to follow to produce reproducible data as well. So before going into that, I'll also like to emphasize the use of CITE-seq over single cell analysis. So we have listed out few differences between single cell and CITE-seq analysis. First and foremost being data modality. In single cell RNA-seq, we are only concerned with the mRNA transcript counts. Whereas in case of CITE-seq, we also include an additional data modality which is antibody-derived tag counts. Normalization done in case of single-cell RNA-seq is log transform and acid transform to normalize the RNA count. We utilize the similar normalization method in CITE-seq to normalize the RNA counts. But for protein normalization, we utilize methods called as DSP and CLR, which Raj will talk in detail in coming slides. Now coming to the important feature of both these technologies, clustering. In case of single-cell RNA analysis, the clustering is done based only on RNA expression. Whereas in CITE-seq, we can either use RNA, protein, or both RNA and protein to do the clustering. This robust clustering helps in accurate annotation of the cluster when compared to the annotation done only with the help of RNA expression. So as I mentioned, like once we have the ADT and RNA files, we can analyze and form a pipeline. So following are the technologies that we can utilize to create a pipeline for CITE-seq. First and foremost, Nextflow. Nextflow is a domain-specific language based on Groovy, which can be utilized for building scalable and reproducible data analysis pipelines. We further use NF-test, which is a unit testing framework tailored for validating Nextflow pipeline components. We also utilize Docker, which enables containerization of software for consistent environments across development and deployment. Once your pipeline is ready, we can then use Secura Tower, which is a centralized platform to monitor, manage, and deploy Nextflow pipelines. Further, we utilize Python and R to do various statistical testing and bioinformatics data processing. Next up, pipeline overview would be taken up by Raj.
Thank you, Nihal. Hello, everyone. I'm Raj, and I'll be taking over from pipeline overview to the end. So basically, for any pipeline that you build, be it single cell or CITE-seq pipeline, anything that has to do with single cell, there will be four components, right? These four components are input check, quality control, pre-processing, and custom analysis. What is input check? Input check is a module just to do all the sanity checks. It is to ensure that before you run processes that are compute heavy, you have everything that is in place. Because once those processes run, you don't want them to stop at that particular moment because it would be detrimental to different processes, right? So that is not what you want. So input check is there. Once the input check has been done and you have the count matrix, that is when you go to quality control. Why do we need quality control? Because while generating the matrix, there are multiple encapsulations that are sometimes not really cells, but they are just artifacts of sequencing process. Now what these can do is they can skew the process, they can skew the results, right? Therefore you don't want to have them in your sample. So what you do, you can remove them using multiple different methods, such as mitochondrial percentage, ribosomal percentage, mRNA counts, protein counts, and stuff like that. Once both input and quality control are done, then we can move to the pre-processing. It is the most basic step. It is the most standard step in all of single cell sequencing, all of single cell data analysis, where you are just basically normalizing the whole sample in order to ensure that you have taken care of all the technical noise and also the randomness, right? So once this is done, you can then move on to UMAPs and stuff like custom analysis, which is where whatever is your targeted area of interest, that is used. So whatever, if for example you want to do a differential gene expression, then we can cluster them together and then do a differential gene expression. If you want to do a region of interest analysis, we can also do that. If you want to do a gene z enrichment analysis and different analyses, there are different ways to do that. So this is the overview of the pipeline. So now, moving on, we'll be talking about AAD normalization. Now, Nihar told you, right, that when we are talking about a single cell RNA sequencing, there are only one modalities. But with CITE-seq, there are two modalities that we have to take care of. One is ADT, the second one is mRNA. However, we cannot use the same normalization for both the technologies because RNA protein is much different from ADT. Sorry, RNA data is much different from ADT data, right? Why? Because RNA data is sparse, it has a lot of zeros, and it is highly variable gene expression. So therefore, the distribution upon which it is modeled is a negative binomial assumption distribution. However, with protein, we do not have this problem. It is less sparse, but the problem that we have is that there is a lot of non-specific binding, which leads to ambient antibody background. Which means that if you look at protein data without context, you will think that every cell is expressing some protein or the other, right? Every cell is expressing a particular protein, is what I mean, right? So that is why we cannot use RNA methods because they do not correct for technical noise. They can distort biological signals, and the assumption for AET data is that they are mixed Gaussian distributions, whereas for RNA it is a negative binomial distribution. So what do we do? We have two methods that are at our pay. Okay, the first is CLR method. What is CLR? It is centered log ratio. How does it work? So basically, for example, you have a cell, right? The cell has a protein. Let us call it protein E. The expression of that particular protein is X, right? So what you do is you take all the proteins that are expressed in that particular cell and then divide the expression of your target protein by the geometric mean of that particular protein. Then you apply a log transformation to that. Therefore, now the protein expression has been centered to the log of ratio. That is why it is called CLR. But I told you, right? There is background correction that needs to be taken care of because there is ambient background. Has that been taken care of this in this process? No, it has not been, right? So then there are other technical covariates that you want. So therefore, in this centered log ratio, background correction is not done, technical covariates are not done, right? Which results in limited clarity when you are separating the positive and negative populations, right? Does that mean we never use CLR? No, CLR is a very basic package. It comes with Surat. Once you install, it will be there, right? And it is very much for very simple data sets in which you want to do quick inspection, right? So you want to do quick inspection, you want to look at the data, you want to understand a broad overview of the data, use CLR by all means. However, if you have like thousands and tens of thousands of data points, it would be better to use DSP. What is DSP? DSP is denoised and scaled by background, right? Now we'll see how DSP is better than CLR. So whenever we are doing single cell RNA sequencing or anything related to single cell RNA sequencing data analysis, what we do is we use cell generation matrix, right? Sorry, matrix generation platforms, right? Which can be like, for example, Cell Ranger. Now, whenever we are using Cell Ranger, it is always told that you should use filtered matrix. However, in this case, we'll not be using filtered matrix, we'll be using raw matrix. Now, what is the difference between filter and raw matrix? The only difference between filter and raw matrix is filtered matrix have lesser artifacts because the blatantly zero emulsions that are there, they are removed by the process itself. However, in this case we will use raw matrix because we want that ambient protein signal that is there in those particular barcodes that have no cells. This will help us to fit the background distribution and normalize. So once we identify the empty droplets and we have fitted it, what we do is we estimate the mean and the standard deviation. Once that is done, we'll use these two variables to normalize for the raw counts. How will we do that? We just take the raw AAT value that is there, just subtract the mean of that background, and then divide it by the standard deviation of the background. This will give you the first step. This will normalize the data for you. Then comes the second step, which is totally optional. If you have the means, by all means go for it. But if you don't have it, do not sweat over it. It's fine, right? If you have particular cell specific regressors that you don't want, like for example, if you have particular isotopes, if your markers have different isotopes that you don't want it to take care of, so you can use isotype no, sorry isotype control levels, right? If there are noise from background features that you don't want, you can do that. Once this is done, you then fit it on a linear scale or a PCA for the background modules, and then you regress out these all of these components from each cell's DSP scores. Once this is done, you get an output matrix, which is Z-scored expression, right? Now this DSP process is complete. What it will give you is expressions that are correct, and we'll see in the next slide how it will work, right? So now, once the normalization is done, you can identify and characterize cell populations using these protein data. Why? Because once you plot these particular normalized ADT counts, you will find that they are multimodal distributions. Multimodal distributions, they'll have two peaks, right? So the peak, that is the higher peak, which is towards the left of the line that you're seeing, that is the background peak, right? What is it? It is the ambient signal of antibodies. It is non-specific binding, right? Whereas towards the right, it is the true specific binding of the antibodies, right? And it represents the positive expression of targeted surface markers, right? So basically we look at this, and this will help, and the value between these two peaks will help us determine the cells which are positive for your target protein, right? Which are there for your target protein. Now this can be used to identify different cell types. There are different methods, there are other methods also, but this is also one of the methods that you can use. So for example, for example, you want to find a cell that is protein A positive, protein B negative, right? Which is protein A, protein B can be your protein signatures. You can obviously look at these two expressions, determine, and then look at the intersection of the cell barcodes, and that will be your target population, right? There are other methods of doing cell type annotation, which it can be single cell, it can be single R and stuff like that, right? But why do we need customized algorithms, right? We need that because existing strategies cannot always correctly estimate the cell population. It is due to the fact that there are complexities that are there in expression data, and they mainly arise due to different upstream sequencing and analysis technologies that are used. So if you use a technology A, for example, if you use different versions, like for example, we have seen if you use cell range 5 and cell range 7.0 versions, you will find different expressions, right? So therefore it is imperative that we develop a custom tailored approach to address these challenges in order to improve cell identification. The effectiveness of this approach can then obviously be verified by results with corresponding wet lab data. And once you have a mix of both dry lab and wet lab data, it is obviously the holy grail of research. So yeah. So now this is done. So moving on, while Nihal was talking about the technologies that we need in order to develop a pipeline, right? He was talking about Nextflow, right? Why are we so bullish on Nextflow? Because it is a workflow management system where you can design scalable, parallelized bioinformatics pipelines, right? What are the main features? The first main feature is that it will give you modularized and plug and play design. So basically, you can have different modules. And for example, if you don't want a particular module or if you want to upgrade a particular module, you don't have to deal with the whole system. You can just change that particular system, like for example, if you want to move from cell ranger 5 to cell ranger 7, you keep everything intact. Just take the cell ranger 5, remove it, develop a new module on cell ranger 7, fit that particular module around that part, and it is good to go, right? It has robust reproducibility and traceability. So what it means is, for example, let's take the four components of the pipeline, right? So the input is there, quality control is there, pre-processing is there, and custom analysis is there. For example, if you encounter an issue at pre-processing, right? Normally, what you would have to do is you'd have to run all over again, which would mean that you'd have to run it for over 15 to 17 hours for matrix generation again, right? In this case, what you have to do is you just have to go there, you have to find out that particular bug, fix it. Once you have fixed it, it will start from pre-processing again. It saves a lot of compute time. You can reproduce your errors, you can trace it, and you can do it at all times. You want to run multiple samples at different times, multiple samples at the same time? Yes, go ahead. Parallel and scalable execution. You can integrate any module that you want, right? So now your pipeline is ready. What will you do with it? You have to test it, right? And with Nextflow, we have a very simple method of testing, which is called NF test. It is a simple testing framework that will facilitate comprehensive end-to-end testing of Nextflow pipeline. The language is also very similar to Nextflow, and you don't have to do anything. You just have to explain the desired behavior using the when and then blocks, right? So what is when and then blocks, right? The when block is basically the place where you will put your parameters. So as you can see, here are two parameters, right? There is only an out there which is where the output directory, and then the second is input parameter 0, which is the input that you'll get. Once you have given your XYZ parameters, then comes the then block where you will just put your assertions. So here we are just asserting for the process to be a success. If you want, you can go up to very granular levels where, for example, if your process was generating two PNGs, right, and you want to test if it is currently also generating only two PNGs. If your process is generating three PNGs, it will specify you that, boss, it is generating three PNGs X in place of two that you had specified, right? So that is the level that you can go. It is very easy to generate. So all you have to do is you have to just type nf test generate and then give the directory path of the particular process, right? Once that is done, it will generate a process for you. And to run, it is just nf test, right? Once it is done, once you do it, if the process is a success, it did not run without it run without any failure and it was done. Apart from this, in the pipeline, you can also introduce different features such as stub run feature, which is just a dry run feature, a dry run feature to just test that all the channels are working properly and none of your compute is being utilized in this process. You can also do CI/CD profiles, which you can test the complete pipeline for, right? So, in summary, we at Strand have the capability to design and implement robust computational pipelines so that we can analyze CITE-seq data in great detail. Right? We can also implement plug and play pipelines that are compatible with diverse count matrices generation platforms that can enable seamless transition and enhance interoperability. There are different engineering novel algor- and we can also engineer novel algorithms for different custom analysis as per the requirement of experiment and study. We can build comprehensive test suites and execute end-to-end validation for different scripts and report all the modules. We can develop CI/CD test datasets with run times just under 30 minutes, so you can test the whole pipeline. without having to do a dry run. And then we can integrate high impact features, including pilot runs, integrative multimodal analysis, automated annotation, and customizable report generation into the pipelines as per your needs. That is all from my and Niha's side. If you have any questions, feel free to drop them. And you can, if you have any other questions, you can contact us at bioinformatics@strands. You can visit our websites us.strandls.com for different case studies. And if you want to get in touch for any future projects. Thank you.
Thank you, Nihar and Raj. That was a wonderful presentation. I would request the audience to please, you know, raise any queries that you have related to the presentation. You can either put it in the chat box or raise your hands. I'll let you unmute yourself and you can have a discussion. Anyone? Any questions? Looks like our audience is quite satisfied with the presentation, and I don't think so there are any questions related to it at the moment. But of course, you can feel free to reach out, and we will also be circulating a form after this, which where you can drop in your queries, and we will direct it to Raj and Nihar. Oh, okay. There's one hand raised, I believe. Someone had raised. Not sure. Yeah, 1 minute. Shri Vidya, yes, you can unmute, please.
Um, Hi, that was a very nice lecture. I just wanted to know, for infectious disease, how can you use your CITE sequencing as in diagnostics?
For infection. So if you're, hi Shivika, thank you very
Hi.
much. So if you're, sorry, hello.
Yeah.
Yeah. So if you're going to use infectious diseases, it has to be first of all, you don't really need to use for infectious diseases because they're pretty transient in the matter. Also, if you're going to use it, one thing that is there with CITE-seq is that it has to have a target that is easily identifiable, right? That has to be
Mm-hmm.
expressed. For example, when we are talking about the different examples that we're using in cancer or blood cell leukemia, we are talking about different markers that are out there, like CD19 marker or CD20 marker. So it has to have a specific, you know,
Mm-hmm.
specific marker that you can target in that particular regard. So, for example, if you're going to use it for common cold, it will be very difficult because every season the common cold changes its variant, right? So
Yeah,
in
no,
that case,
actually, especially these like fungal infections and your bacterial infections, they have a like specific set of TLRs which are recognized by the host cell.
yes.
So can
So
we
if you
use that?
yeah, you can design that, but do you want to do it for a infectious disease is the question. In that sense that it is a very expensive technology, right? So
Okay.
you have to also look at the cost of logistics that are involved because you are taking out the blood from a person doing a clonal expansion, then putting it back into the human being, right? With cancer, you know that that particular thing will persist for a really long time, and that will not change its signature for that much, right? With
In that
it will,
sense, I think for autoimmune diseases, this will be helpful.
yeah, you can do that. But
Okay.
again, but again, yeah, it's there's a component of cost, which is because this is a very expensive technology. Uh, the site seek process in general, the data analysis is not.
Oh, thank you.
Yeah.
Thank you, Shri Vidya. Anyone else? Any queries? Okay. So thank you so much, Raj and Nihar, for that wonderful presentation. And on behalf of the entire Strand, I thank both of you to a sincere gratitude to both of you for coming up and doing this presentation. Thank you so much, and I thank our audience for staying for so long. And yes, thank you all. We will be circulating a form where you can please feel free to reach out to us and drop in your queries, and we'll get in touch with you. Thank you so much, everyone.
Thank you, everyone.
Thank you, everyone.